Hello dear students, I hope you all are doing good. I'm Miss Imkongmla from the Department of Biology, and today we will be learning about the chapter Strategies for Enhancement of Food Production. Now, as we all know, population is increasing day by day. As a result, the demand of food requirement is also increasing as well. So in order to fulfill this demand of food requirement, different strategies are required to enhance the food production. So in this chapter, we will be learning about the different strategies which will help in increasing the food production. Now the strategies involved are animal husbandry, plant breeding, single-cell protein, tissue culture. Now, what is animal husbandry? Animal husbandry means breeding and raising livestock. Livestock includes animals like cows, buffaloes, sheep, goats, camels, etc. Now, under animal husbandry, again, there are 4 types: dairy farm management, apiculture, poultry farm management, fisheries. Now, what is dairy farm management? It is the management of animals for milk and its product. For milk and its product. Now, what are the parameters which we have to keep in mind? Now, the first parameters for dairy farm management is selection of good quality breed and well-maintained house with proper hygiene and proper supply of water. And the most important thing, regular visit of veterinary doctor should be mandatory. Now, what is apiculture? Apiculture is the rearing of honeybee hives for honey production. Now, the most commonly reared honeybee is Apis indica. Apis indica. Now, poultry farm management. It is the management of fawn. That is birds, like example chickens, duck, turkey, etc. for their food, that is in the form of their meat, and for their eggs. Now, about poultry farm management, some parameters which have to be kept in mind is selection of disease-free breed, proper supply of water, well-maintained hygiene, and the most important, artificial light should be provided, because artificial light helps to increase their weight and also their egg production. Now, about the fisheries. It includes rearing, catching, processing and selling of fishes, shellfish or aquatic animals. Example of some freshwater fishes are katla, roho, I'll write it here, katla, roho, carp, etc. Now some examples of marine water fishes are pamphret, hilsa, magaric, etc. Now, what is blue revolution? Blue revolution refers to the increasing production of the fish. Okay, now, once we are done with the animal husbandry, the next important topic will be animal breeding. Animal breeding is the mating of two similar or dissimilar breed. Similar or dissimilar breed. Now, what is breed? It is a group of animals having related by descent or having common characteristics like general characters, appearance, size, configuration, etc. Now, under animal breeding, two types. The first one inbreeding and outbreeding. Under outbreeding, again, three types. Outcrossing, crossbreeding, and interspecific hybridization. Now, what is inbreeding? Inbreeding is the mating of more closely related species with the same breed. Now, that is for 4 to 6 generations. Now, suppose example, if we take a superior female cow and it is mated with a superior male bull, then the expected offspring will be calf. Suppose female calf. Now this female calf which is produced is allowed for another mating with the same bull. Now continuous breeding will go on. As a result, suppose in the first mating we get 10 liters of milk production and in the second 6 liters. Say 8 liters, 6 later off. So as per the continuous breeding or mating goes on, the milk production will decrease, like 10 liter, 8 liter, 6 liter, because here lies the disadvantage of inbreeding, which is called the inbreeding depression. Inbreeding depression means it reduces the fertility and also the production. Now, under outbreeding, outcrossing. Outcrossing is the mating of unrelated species of the same breed or between the same breed. And crossbreeding, it is the crossing of one superior male of same breed with the same breed with another superior female of another breed. That is crossbreeding. Now, interspecific hybridization. Interspecific hybridization is the cross between two different species. That is, two totally different species. For example, a male donkey is mated with a female horse. That means two different species which produce offspring, a mule. This is the best example for interspecific hybridization. Now the next topic, controlled breeding in animals. Here the first is artificial insemination, AI, and the second one MOET, that means multiple ovulation and embryo transfer. We will discuss one by one. The first one, artificial insemination. It is a technique where the semen from a selected male will be collected and it will be injected into the selected female. That means into the reproductive tract of the female. Now the semen, after collection, it can be directly incorporated into the female tract, or it can also be stored in frozen form for later use. Now the second one, moit. Under moit, as the name suggests, here multiple eggs will be fertilized in an animal and the embryo production will be collected, after which it is one of the traditional method where the embryo production is being done, and it is practiced in cattle. Now in this process what happens is the cows will be administered. The cows will be administered with FSH, that is follicle stimulating hormone. That means the cows will be administered with FSH hormone. Now what this hormone does is it induces follicular maturation and superovulation in the female. Now as a result, after 7th day, the eggs, that means the embryo, will be collected and it will be transferred to the surrogate mother for further development. Now here in this process what happens is the gauze which is administered by FSH will be able to produce 6 to 8 eggs instead of 1 egg per cycle. That is the advantage of this moid technique. Okay, the next is plant breeding. Plant breeding is the manipulation of plant species to obtain a desired plant type. Now, why plant breeding is done? It is done to increase the crop yield. It is done to increase the tolerance to environmental stresses like salinity, extreme temperature, drought, etc. And also to increase resistance against pathogens attack. That is, by the attack of virus, fungi, bacteria, etc. Now there are different steps involved in plant breeding. The first one, collection of genetic variability. The second step, evaluation and selection of parents. Number 3, cross hybridization of selected parents. 4, evaluation and selection of superior recombinants. And the last step, number 5, testing, release and commercialization of superior cultivars. Now, collection of genetic variability. Now, in this step, what happens is we have to collect different wild species of plants. That is nothing but germplasm collection. And in the second step, we have to evaluate the plants which we have selected, which we have collected in the step 1. And we have to choose, that means we have to select the parents with the desirable characters and then multiply it and use it for hybridization. Now in the cross hybridization third step, the selected parents will undergo hybridization with the help of emasculation and packing method. Now once that is done, after cross hybridization, hybrid plants with common characters of both the parents will be produced. Now in the step 4, the hybrid which produced will be further evaluated for their high yield or for their quality or for their disease free. Now this evaluation is done in research fields, at least for 3 generations. And at last, finally, here in the testing, release, commercialization will be done of the selected hybrids and it will be marketed to the farmers. Now these are the 5 steps which you have to remember under the plant breeding. Green Revolution. Green Revolution is the development of high-yielding wheat and rice varieties. Wheat and rice varieties in 1960s with the help of plant breeding techniques to enhance the food production. Now, Green Revolution, two high-yielding wheat varieties found in India are Sonalika and Kalyan Sona. And 2 examples of high-yielding rice varieties are Chaya and Ratna. Now, another example of plant breeding techniques can be of sugarcane also. Sagaram Parbury, Sagaram officinarum. These 2 are sugarcane species. Now this is found in North India, which has a very poor sugar content and poor yield. Whereas Sagarum officinarum is found in South India, which has thicker stems, it has high sugar content and also high yield, but it cannot be grown in North India. Now with the help of plant technique methods, these 2 sugarcane species were allowed to undergo cross breeding, and as a result, the hybrids obtained after cross breeding has thicker stems, higher sugar content, and high yield. And most important, has the ability to be grown even in North India. So this is also one of the example under plant breeding advantages. Okay, next is biofortification. Biofortification means the development of crops with higher levels of minerals, vitamins, proteins, and healthier fats. So there are two examples. Atlas 66 is an wheat variety which has high protein content, and the second example is iron fortified rice which has rich iron content. So under for biofortification, these two examples are important. Okay, now next is single cell protein, SCP. Single cell protein is an alternative protein source of nutrition which is extracted from useful beneficiary microbes, and the microbes are spirulina, chlorella, and methylophilus. Now these are the three examples of single cell protein. Now tissue culture. Tissue culture is a technique of culturing cells into tissue, organ, or organism on culture medium under laboratory conditions. Now, what are the components required for tissue culture? The components required are vitamins, distilled water, minerals, amino acids, sucrose, growth hormone, and agar. So these are the components which will be required to prepare a culture media. Now, what are the procedure in the tissue culture? Now, the first, from a healthy plant, any part of the plant body that is called explant. Any part of the plant body that is called explant will be taken. It may be from the leaf part, it may be from the stem part, any part. It will be cultured in a petri dish. Explant will be cultured in the petri dish, in the petri dish or viticulture medium is incorporated. Now this will be followed by this eggplant will form callus. Callus is an undifferentiated and unorganized mass of cells. Now in the next step, this callus will be separated as single cells. It will be separated as single cells and it will be cultured. Now, further culturing will finally generate a new plant. Into a new plant. This is the steps involved in tissue culture. Now, what are the applications of tissue culture? The applications are production of large number of plants from a single cell or from a small mass of tissue, and production of genetically identical parents plants, that is called soma clones. And seedless plants can also be multiplied. An important thing, recovery of healthy plants from a diseased plant with the help of meristem culture. Let's discuss a bit about the meristem culture. Suppose we have an viral infected plant. Now we want to get a disease-free plant from that particular infected plant. Now what we do is we take the meristem part from the infected plant, and that meristem will now act as the explant for the tissue culture procedure. Like we take the petri dish and the meristem is cultivated. Now from the meristem, callus formation will take place and callus will be separated, the same technique of tissue culture. So this is how from a virus infected plant we get a disease-free plant by taking the meristem as the explant. Now what is totipotency? Totipotency is the ability of a single cell to develop into a tissue or an organ or an organism. Now micropropagation. What is micropropagation? Micropropagation is the process of producing thousands of plants with the help of tissue culture. And the next is somatic hybridization. Somatic hybridization. Somatic hybridization is the fusion of protoplast of somatic cells of different species of plants to produce somatic hybrids. Different species to produce somatic hybrids. Now, the best example for this is the fusion That means the fusion, protoblast fusion of potato and tomato to form tomato. Now class, that's how this chapter ends. If there is any inquiry, kindly feel free to call me to clear your doubts. Thank you.